This tutorial is about data analysis in the Baxter algorithms. Here I have opened a dataset with 115 image sequences. The image sequences have muscle stem cells cultured on either tissue culture plastic or on hydrogel. This is a microwell with a diameter of 600 microns, and this is a muscle stem cell. The images were taken with 10x magnification, and the time between images is 3 minutes. In order to perform the analysis correctly and to get distances in micrometers and time intervals in seconds or hours, first need to specify settings for the image sequences. So you select the analysis category, and here you see that I've entered 6.45 microns for the pixel size, and this is the pixel size of the camera sensor. And here's the magnification, that's 10x, meaning that we have a pixel size in the images of 0.645 microns. And then I've also entered the time between images, which is 180 seconds or 3 minutes. I also specify the conditions, the culture conditions of the two experimental conditions. So here is tissue culture plastic, and then further down I have image sequences with a gel substrate. So these are the settings that you need to enter before you can do the analysis. In order to do the analysis, you also need to track the cells. I've explained how to track cells in earlier tutorials, so I expect that you've already figured that part of the processing out. So once you've done that, you can go to Analysis, and the first two options here are for analysis of tracking results. So the first one, Cell Analysis, Graphical User Interface, is a user interface where you can look at cell properties of individual cells over time, so you can watch different parameters, how they evolve over time. And this option will take time averages over the cells and analyze the entire population of cells. So I'll open this one first. Here I select the tracking results that I want to analyze. Here I'll take the tracking results called core. They've been corrected. The tracks have been corrected, but the outlines of the cells have not been edited. Here you can look at a number of different parameters of the cells. The first parameter here is the axis ratio of the cells, so the ratio between the length and the width of each cell, more or less. A value of 1 means that the cell is close to circular, and larger values means that the cell is stretched out. So the first cell of this clone starts out round, and then it starts to stretch out. And then just before it divides, it becomes round again. Then you can watch how the daughter cells, so the progeny of this initial cell, evolve in terms of axis ratio. Here you can select to have the time scale either in frames or in hours. So it's 35 hours or about 650 frames. If we look at multiple plots, we see that the y-axis changes over time. If you don't want it to change, you can set a maximum value it's a 6, for example. And then the axis ratio scale is from 0 to 6, no matter what the values of the individual cells are. The next parameter here is the lineage tree. So we can look at the lineage trees that we see in the correction user interface as well. And here we can choose between analysis per image or per clone. So if we have multiple starting cells, each starting cell is considered a clone together with its progeny. So here is one image sequence with 2 starting cells. If we select images instead here, both of these starting cells, they don't have any progeny in this case, but both of these lineage trees are plotted in the same axes. And same thing is true if we plot any of the other parameters. So the next parameter is the cell size, and if we have something with a length unit or an area unit on the y-axis, we can choose between pixels and microns. So here it's in square microns, and here it's in pixels. And we can still choose between different units on the time axis, of course. Here on the, we can go through all of them. So we have the cell count, that's the number of cells in each image sequence, the number of live cells. And then there's total distance traveled, and that's the total distance traveled during the experiment by the cell and all of its ancestor cells. So here we can see that the starting cell has a constant velocity. Then the 2 daughter cells, the green cell here is slightly faster than the purple cell. When cells divide, we can see that the mother cell slows down, and then the daughter cells are slow in the beginning. If we want to look at multiple plots, we can set the frame rate here to 2 frames per second or something like that, and then we can have the program go through the image sequences one at a time. And the 3 following parameters are lineage trees, where the y-axis is some parameter. In this case, it's the average velocity of each cell. So here we can see that the starting cell had an average velocity of a bit more than 60 micrometers per hour, and then the 2 daughter cells have velocities of 90 and 125 microns per hour. And then the 3rd generation speeds up even more. And the following parameter is the axis ratio. So we can see here that for some reason the cells become more and more round from the 1st to the 2nd to the 3rd generation. And here's the average area. And this is only for one image sequence, of course, so you would have to look at the entire experiment and see if you can see a trend in the data. And the final plot is the trajectories of all of the cells during the entire experiment. And here you can see migration patterns, and you can also see if the cells stay in a small part of the microwell, and that can be an indication that the coating of the surface with a protein that the cells can adhere to was not successful. But these trajectories don't indicate that. If you want to save these plots, you first select the plot type that you want to save. If you want to save a single plot, you go to Export, Export Plot. Then only the displayed plot will be saved. Here you select where to save the plot, and I'll do it on the desktop. And then you select a file format. The PDF doc will create a PDF where each plot has a separate page, and there's also a title page. And then Tesh will create a LaTeX document that can be compiled into that PDF document. And if you want to make changes to the PDF documents afterward, you should save the Tesh document as well. Otherwise, that's just removed. And PDF and EPS, those are two vector graphics formats where each image, each plot saved as a separate image. TIFF will create TIFF files with pixel images, and FIG, that's MATLAB's figure format, so that you can open the images in MATLAB again and change line width and stuff like that afterwards. That format can't be used to import into documents and stuff like that. But I'll save a PDF document, and I do it on the, I think I'll do a TIFF instead. I can save a PDF document later. And you can set the height and the width and the resolution as well. And you can select different plotting styles depending on what you want to use the saved document for. If you select none, the plot that you see here will be saved as it is. So then I press save, and my TIFF image appears on the desktop. Now if I want to save multiple plots, all of the plots for the different image sequences, then I go to Export, Export All Plots, and I choose to save this on the desktop as well. And I save this PDF document. The title will be axis ratio. I can edit it if I want, and I'll enter the author string. It's my name. Here you can use LaTeX syntax, so double backslash is a new line. And I'll choose to save it as a letter-sized document. When I press save, the program will start to save PDF documents for each of the individual plots, and once it's done doing that, they will be compiled into a single PDF document. In order to do this, you need to have LaTeX installed on your computer, so this might not work for you. But you can still save the PDF documents, the individual PDF documents. So now, here's the compiled PDF with all of the plots. And this can be very useful if you want to send your results to someone else through email. And the third option here exports an overview plot, or it generates figures with overview plots, so that you can look at multiple plots at once on the same page. And to save these, you need to go to the main user interface and go to Analysis, Save Plots. Open the user interface for saving of plots. And I'll deselect all of the plots that aren't overview plots. And then I choose to save it on the desktop. And I save a PDF document again, but I'll change the title to Overview of Axis Ratio. I'll just enter my name as an author to the document. And then here is the PDF we just generated. So this was everything I was going to show in the cell analysis user interface. You can actually close all of the figures from the main user interface. And then I'll show you the population analysis user interface. And here I select the tracking version that I want to analyze. I'll select core, just as in the previous interface. And here you can look at time averages for each cell. So each cell here is a dot in the scatter plot. You can look at the average speed of the cells, the average axis ratios, average size, the time between division, the difference in time between division between different daughter cells. So when both daughter cells divide, you can see the time between their divisions. And time to death is the time in hours until the cell dies. And only the cells that die are marked here. If we look at the average speed again, there are a number of different plots that we can look at. We can look at the scatter plot. We can look at the histogram, kernel smoothing density. That's sort of like the histogram, but it's smoother. Just the sorted values, or a CDF, cumulative distribution function. We can look at how the parameter is distributed among daughter cells. So this is the speed of the slower daughter cell in sister cell pairs, and this is the speed of the faster daughter. You can see that there's definitely some correlation between the speeds of the daughter cells. And we can also look at clones of cells, where the mother cell is on the x-axis and all of the daughter cells are on the y-axis. And so this one shows all of the progeny of a single starting cell. And here is the same thing for only one mother cell and the daughter cells of that mother cell. And we can also plot this parameter over time to see how the average speed of cells changes over time. And the time here is the time when the cell divides, or when it's created through cell division. If we go back to the scatter plot, we can choose between these three checkboxes. So separate will separate the cells by cell generation, so we can see that the first generation is slower than the second generation and even slower than the third generation. And if we mark dead cells, the dead cells will be shown in red. We can also mark cells that survived to the end of the experiment. So these blue cells are cells where we haven't seen the entire lifespan of the cell. So that could be a reason why the cells are slightly slower here, because they're slow just after they've divided or been created through cell division. Here we can save plots. We can actually mark multiple plot types here, the average speed and the average axis ratio. If I press save plots, I get to choose where to save it. I'll save it on the desktop as usual. And I will save a PDF document. Enter me as an author. And press save. So then I get a PDF document like this with the two plots that I selected. I think that's everything I was going to show in this user interface. A third part of the analysis is the statistics menu, where you just save statistical readouts for the individual cells to an Excel document. So I select the tracking version that I want to analyze, and I select where I want to put it, and then the program will save a PDF document with time averages of different cell parameters. Now we can open the Excel file. It contains a lot of different data, but here I'll look at the average speed of the cells. So in tissue culture plastic and gel, we had 309 cells in tissue culture plastic and 222 cells in the gel condition. And the mean cell velocities were similar, slightly faster on the tissue culture plastic. And here they've been separated into cells that died and cells that didn't die, and also into the different cell generations. So the first generation is the cells that were present at the beginning of the experiment. The second generation is the progeny of those cells, and so forth. So we can see that the first generation was a lot slower than the other generations.